The other day, someone expressed confusion about the term expression. And you know what, folks? I feel ya. This is a really weird term to try to explain and describe because there's a lot of nuances. But when we're talking about expression, we're typically what we care about is a functional product being made productively. So like if in a certain cell is a protein that you're interested in being made, or is a functional RNA being made. But there are different levels of regulation that can occur throughout that making process, and so if you measure expression at various steps along the process, then you can get insight into different things that might be going on, and so there are different ways to get different snapshots of expression. So let's take a look at some of the levels of gene expression regulation and experimental measurements. Expression. It can be very hard to express what expression means because it can mean different things in different contexts and there's a lot of nuances. So let's try to get to the heart of what we're talking about. The genetic instructions for making different products are called genes, which are stretches of DNA that are part of longer pieces of DNA called chromosomes. So a gene is like a stretch of the chromosomes, so kind of like a chapter or something of a big book. And it has the instructions for making a product. Often these products are proteins, and that's what you usually hear them referred to, so that genes are like the recipes for making proteins. But that gets difficult when you talk about expression because not all genes actually have the instructions for making proteins. Some genes make things called functional or non-coding RNAs. So we talk about coding RNAs, we're saying that they code for a protein, so they have the instructions for making a protein. But there are also RNAs that function as RNAs instead of as just intermediaries between a gene and a protein. So we're going to talk mainly about focusing on the protein making things here. But for expression, what you're typically trying to get at is whether that final product is made, although that's not always how you're measuring things. So if you're looking for a functional RNA being expressed, that would be is that functional RNA expressed? Is that final product the RNA made? If you're talking about a protein, you're probably most interested in whether that protein is made. But that involves going through an intermediary mRNA step, and so often that mRNA step is what's measured when you're talking about expression. So when you're talking about gene expression, you can either be talking kind of at the level of is the mRNA being made, or is the protein being made? But ultimately what you usually care about is whether the protein is actually in the cells. So then you also have to take into effect things like modification and degradation. So we'll get into all of these and some of the different ways that you can measure these different things. So we have this general, we're going to focus mostly on the proteins because it's easiest to explain and because it's often what we're talking about in biology. Even though some of the coolest things are non-coding RNAs like microRNAs, which I have slides on and which I talk about in that study. So basically you have a gene which is written in DNA, and so that's that stretch of the chromosome, so that portion of a longer piece of DNA that gets copied as a unit into RNA form. So this is your messenger RNA, and that gets edited, so it gets these regulatory regions called introns removed in a process called splicing, so you get this shorter mature messenger RNA or mRNA. And then that mRNA gets used by the protein making complexes called ribosomes to make the protein, which could then get modified and degraded. So it might seem weird to have that extra step of making the mRNA copy of to be translated, but this actually allows you to protect the original gene copy as well as make lots of mRNA copies so that lots of chefs can be working on it at once. It's kind of like if you have the bakery where they lock up that original recipe, but they make copies of the recipes that they want the chefs to make when they want to make them. So maybe if in the summer you'll have like fruit pies, but then in the winter when those fruits are out of stock, you'll have like canned fruit pies, and then also have different recipes. So you can make different recipes at different times. And you can make more or less depending on demand by making more or less copies of the mRNA and or regulating the levels afterwards. Another reason that we have the mRNA step is that in our cells, the genes are housed in membrane-bound compartments called the nucleus. And so the gene to RNA process is called transcription, and that takes place with the protein called RNA polymerase. So it's a DNA dependent RNA polymerase, so it takes a DNA template and makes an RNA copy. This transcription happens in the nucleus, and then the mature messenger RNA is exported out into the general cellular interior called the cytoplasm. And that's where the translation, so the protein making, occurs. So having that RNA step also allows us to keep the DNA separate in the nucleus. That's in our cells. But last week we were talking about bacterial overexpression. So this is basically where we take the DNA form of the mRNA called cDNA. So basically the copy of the genetic recipe. We stick that into a circular piece of DNA called a plasmid, and we stick that into bacteria cells. The bacteria cells don't have the nuclei, so all this is happening in the cytoplasm. And there tends to be a more direct link between the mRNA and the protein making, so there's like transcription and translation are tightly coupled, and we don't have to worry as much about regulation post-transcriptionally. So we're often when we're talking about recombinant protein expression, we're usually talking about we're making the protein in the cells. So let's talk a little bit more about regulation, because that's a big thing in our cells. So you can have multiple levels of regulating expression. The first level is transcriptional regulation. So this is kind of like turning on the fire hose. So this is the most dramatic kind of effect, but it has the longest time to take effect because you're cutting off the source, but you still have all the that like water to clean up in our fire hose analogy. So with transcriptional regulation, you're talking about how many mRNA copies do you make. This depends on things like transcription factor proteins binding, as well as epigenetic modifiers. So when we talk about DNA, so chromosomes, what is a chromosome really? It's this like double-stranded DNA that's really, really coiled up. And this coiling up is done with the help of these proteins called histones. So it's kind of like beads, like wrapped around. It's often explained as like a beaded chain type thing, where you have these histone, these octamers, so these like 8 little proteins which have the DNA coiled tightly around them. And these histones have these tails, so there's like part of the protein, the ends of the protein like stick off and wag around, and they can get modified by things like methylation and acetylation. So basically these extra chemical groups get added onto them, and that affects how tightly the DNA gets wound. So we call this these like epigenetic modifiers. They're above the genome, the level of the DNA. So they're not modifying the DNA directly, but they're modifying how the DNA is like wound up and how it can be accessed. So when it's more easily accessed, then the RNA polymerase can make copies of it, so you get more transcription. Proteins called transcription factors can also bind to specific sequences in the DNA to promote the recruitment of RNA polymerase or chromatin remodelers, or to suppress the expression of the gene. So that's transcriptional regulation. But after you make the mRNA, you can still have regulation. So now you can have regulation, post-transcriptional regulation. So this can depend on like how long those mRNAs stick around. So in general, this can depend on things like how long the poly A tail that they get is, how the presence of binding elements, and if you want to have like specific degradation of specific mRNAs, there's this thing called microRNA or RNA which is a kind of RNA interference, where small RNAs called microRNAs or siRNAs can be used to, they have like sequences that match to sequences in the 3' UTR, so like the end region of the mRNA that's past the protein coding part. So like a regulatory region at the end. These microRNAs have sequences that match sequences in those mRNA targets, and they guide destruction machinery to shut down production. And so this is actually the topic of my research, so I have lots more on that on my blog and in my past posts, so I won't go too far into that, but just know that it exists and it's really cool. Then so that was at the level of the nucleic acids, so the DNA or the RNA. But now I want to talk about translational regulation. So how many proteins are made from the mRNAs? And so this is going to depend on things like the sequence features, the codons, etc. So basically in translation, what happens is your mRNA, it has the protein instructions are written in the form of like 3 RNA letters form or spell one protein letter. And so we call these 3 mRNA letter words codons. And so this protein making complex called the ribosomes travels along the mRNA, and it goes codon by codon. So it's like going in 3 letter chunks. And so like each chunk that it gets to, it has another molecule called a tRNA, which is one of those functional RNAs. So it's an RNA that functions as an RNA instead of being just an mRNA intermediary. So this tRNA, on one end, it has an anticodon that matches the messenger RNA's codon that it's sitting on. And on the other end, it has the corresponding amino acid, so the protein letter. So it comes and it puts that down, and then the ribosome moves along. There's different tRNAs, so sometimes protein letters have multiple codons that spell them. And so they can have multiple tRNAs that bring that same amino acid. Sometimes cells have this thing called codon usage bias. So organisms have like codon usage bias, where different organisms prefer using different codons that spell the same amino acid, so they'll have more of the corresponding tRNA stocked up. So what you can have is this thing called like rare codons, where there's not many tRNAs of that, or you can have a case where you have lots of the same letter in a row, and so there could be like a temporary tRNA shortage while you're trying to go find a tRNA that matches. And so this can cause things like ribosome stalling, and it can affect how quickly a protein gets made and how much of a protein gets made. So that's just one example of a sort of translational regulation, but there are also others, including protein binding factors. Okay, finally you have post-translational regulation. So this is about like how long do those proteins actually stick around. So it's one thing to actually make the protein, but if you make it and it just like disappears right away, that's not very useful. Like, so technically your protein is expressed, but it's not like, it never gets a chance to actually do its thing. But usually what happens is the modification or degradation happens later on, so the protein still gets to work. It just gets degraded once it's no longer wanted. And so how this degradation takes place is usually through this process called the ubiquitin proteasome system. So it's a different kind of UPS than you're probably used to. But there are sequences called degrons, which get recognized by these ubiquitin ligases, which stick this molecule called ubiquitin, this little protein called ubiquitin, onto the protein, and this serves as a sort of tag that gets bound to the proteasome, which is like this protein chewing complex, and then the protein gets chewed up. So we have all these layers of regulation. And so when you go to measure protein expression, depending on which step you measure, you're going to see different things. So you're going to see the effects of different things. The gene in a DNA is transcribed into messenger RNA and translated into protein. So here's a basic overview of some of the ways that we can measure. So we can measure at the transcriptional level, we can measure at the mRNA level, we can measure at the translational level, we can measure at the protein level. So starting with measuring transcription. And so one of the ways that we can do this is with Pol-2-Trip-seq. IP stands for immunoprecipitation. And when we hear immunoprecipitation, think of like antibodies being used to pull out and isolate things that something is bound to. Okay, that sounded really generic. But so antibodies are these little proteins. So we think about them often in terms of antibodies being made against viruses and bacteria. But antibodies can really be made that can recognize anything. So antibodies, the ones that we use are often these like Y-shaped antibodies, where on the ends they have unique parts that recognize the thing of interest. And these are often made using like lab animals and or like expression cells once you isolate a certain the certain gene for the antibody that you want. So with an co-immunoprecipitation, what you do is you use that antibody that sticks to a specific protein that you're interested in, and then that protein, if that protein is stuck to other things, then when you go to isolate the thing that the antibody is bound to, you'll also see what that protein that the antibody was bound to. So if you do this with the RNA polymerase, then you can see what if you like cross-link the RNA polymerase to the DNA that it's bound to, then what you can do is you can use the antibodies against the RNA polymerase to isolate the RNA polymerase and the DNA that it's bound to, and then sequence the DNA that it's bound to see where it was bound. And there are also other things you can do, like ProSeq and GrowSeq, which serve to measure more active promoters or more actively working RNA polymerases, and there's various protocols to do different things. But the basics is you're working at the level of transcription. Next, we can talk about measuring mRNA levels. And so we looked in detail before at one of the ways to do this, RT-qPCR. So the basic idea is that when you're looking at mRNA levels is that the more mRNA copies you have, the more potential you have for making protein. And so RT-qPCR is a way to measure mRNA copies by reverse transcribing the mRNA into a complementary DNA, cDNA. And the purpose of doing this is it stabilizes it and it allows you to use PCR to make lots of copies of it. And so you make lots of copies of it, and then you measure how many copies are made to get an idea of how many copies you started with. qPCR replaced in a lot of ways this technique called Northern blot, although Northern blot still does have specific uses, like if you want to look at modifications. With the Northern blot, what you're doing is you're separating the RNAs, and then you're using oligonucleotide probes to check for the presence of specific RNA. So it's a lot more cumbersome than a qPCR and low throughput. Speaking of throughput, there are more high throughput ways like microarrays, where you have it's kind of like doing a ton of qPCRs on like a big plate with lots of wells, where each one is like doing a different reaction. And then there's RNA-seq, where you're measuring like all the RNAs, just by sequence. So each has pros and cons. But the basic idea there is that the level of mRNA is being used as a kind of proxy for the amount of protein being made. But of course, the amount of protein that gets made is going to depend on translation. So you can actually do things to measure translation directly, including ribosome footprinting and ribosome sequencing and polysome profiling. So I post on these, I won't go into too much detail, but basically it's looking at where the ribosomes are in the case of ribosome footprinting, and how many ribosomes are on each mRNA in the case of polysome profiling, which is used because mRNAs that are more actively being made are going to have typically have lots of ribosomes on them. So we call those polysomes. And so mRNAs that are being actively made are going to typically be in the polysome fraction versus the monosome fraction, which only have one ribosome on them. So you make the protein, but then you can also the protein can also get degraded. So if you want to look at how much of the actual protein is there, you can use methods like mass spec, which mass spectrometry, which basically breaks the protein up into a bunch of different pieces, and then analyzes the mass of those to charge ratio of those pieces. So basically, it's kind of like putting things through a paper shredder and then looking at the shredded paper and trying to figure out what the pieces were. That's not a very good analogy, but you break the proteins up into these different pieces, and then you try to identify the pieces and see what proteins were present. And so it might sound like, why don't you just identify the proteins directly? But that's actually a lot harder with these techniques. So you can actually get it's actually easier if you break the protein up a lot. So it's too technical to really get into detail here. But I will talk more about the Western blot. So basically here what you're doing is using antibodies, so those little proteins we talked about before, except here you're using antibodies that instead of being specific for RNA polymerase, they're specific for a specific protein that you're looking for. And so you separate the proteins using SDS PAGE, gel electrophoresis, which we've talked about, and then you transfer the proteins onto a membrane and you block the membrane using these antibodies to see if the protein is present. And so that's basically a whirlwind overview of a lot of different concepts, and I just kind of wanted to give an overview of why it's so hard to define expression. But so if we're talking about like a protein making gene, we're often talking about like whether the protein is made. But it's not that cut and dry when we talk about expression. And basically, so if you're talking about like a functional RNA, like is that functional product made? So you can think of it as like whether or not a gene is being used productively, if that makes sense. If that doesn't make sense, I'm sorry, and I'm sorry if I confused you more. But this is just something to keep in mind when you're thinking about what expression actually means and why I am having a very hard time explaining what it actually means. But for most intents and purposes, if you're talking about things like recombinant protein expression, what you're talking about is like, is that protein being made? So when I talked about expression in bacterial cells and whether or not the protein was being expressed, is the protein being made is basically what I was getting to there. So sorry if I confused anyone, and hope that helps some people, and happy expressing